Everybody, I'd like to welcome you all to our exit seminar today. And so we have the first of the ARC CCeMMP cohort students to finish, and that's Isabella Russell. And Isabella's had an extremely productive PhD. Currently, she has a published first author publication as well as a co-authored manuscript. This morning, formally accepted first authored review in Pharmacological Reviews. And it's always nice to get those emails. As well as multiple papers that will come from the rest of her thesis. One of the features of the ARC CCeMMP program is that there's an opportunity to do projects in partnership with industry, and also that the students can do embedded placements with industry. So Isabella has been fortunate to be able to be working together with scientists at AstraZeneca based in Cambridge. And she also completed her embedded placement in Cambridge. That was also a productive time for her, including work on multiple projects, part of which was crossing into the dark side, do some crystallography. And again, there's a whole range of papers that we expect to arise from that work in the future. Being an industry program, the timing of those publications will be up to them. And finally, as well as being a tremendous member of the laboratory, Isabella already has a job lined up working with Katerina Toporova, based in Oxford. So congratulations all the way. And with that, I'd like to invite Isabella to deliver her seminar. Thank you.
I think everything's unmuted. I'll just turn this on, which appears to be working. Okay, well, hello. Thank you all for coming. I'm really excited to talk you through some of the stuff we've been up to for the past 5 years, I guess. Without much further ado, I'm going to start by introducing GPCRs. So GPCRs, or G-protein coupled receptors, are a superfamily of transmembrane proteins. There's about 800 in the human genome, and they can respond to pretty much anything you can imagine. However, they all have one unifying feature, and that's that they have 7 transmembrane spanning alpha helices. And once they're bound to a ligand, they'll canonically act through their heterotrimeric G proteins, which is how they get their name, and then have a range of downstream responses. Because there's so many of them, it follows that they're very much involved in both our normal physiology and also disease, and this is one of the reasons they're a prime drug target. And the reason I'm here to talk to you today is because Cryo-EM, the technique I'll be talking about, has really enhanced our understanding of GPCRs. Now, as there are a lot of GPCRs, you can imagine we've come up with different ways to class them. So today I'm only going to be talking about 2 of the many classes of GPCRs. So I'll be talking about class A and class B1 GPCRs. And every time I show you a receptor today, we're going to look at the extracellular side up here and the intracellular side down here, so just to orient you. Now, briefly, just to introduce what these look like, we'll have a class A receptor on the left, your left, and a class B receptor, B1 on the right. And you can see they have a lot in common. So they all have 7 transmembrane spanning alpha helices. They have intracellular loops, extracellular loops. However, the main difference between the 2 is what I'm highlighting here in blue. And that's that all class B1 GPCRs have a large ECD. However, most class A GPCRs do not have one. And class B1 GPCRs use to bind to peptides. So one of the reasons that we've had a bit of a boom in structural biology of Cryo-EM is because we're able to harness what the GPCRs are naturally doing in the cells already, and that's the G protein activation cycle. And because it's quite important to my thesis, I'm going to take it through you, although I'm sure you're all familiar. So we start with our receptor alone in the cell. So this is called our apo form. And you can see here it's a class B receptor because we've got this nice big ECD. Now a ligand eventually will come along and bind. So here I'm showing an agonist as a peptide because it's a class B GPCR. When this peptide binds, it triggers a range of responses in the receptor, touching key motifs that allow transitions to happen. Now these transitions allow our G proteins to bind. Our G protein's job isn't actually to bind to the receptor. Our G protein's job is to turn over GDP for GTP. So when they form a heterotrimer, we can see they do that here. They'll exchange GDP for GTP, and then they go on to dissociate not only from the receptor, but also from each other. So G alpha will then go and do some downstream roles. Today I'm just going to be talking about GS, and the main role of GS is to couple to adenyl cyclase and make a lot of cyclic AMP. So this is useful for us because we can detect activation that way. And then the beta gamma subunit stays together, and they can have a role of downstream. They activate a range of downstream things, including GERK. Eventually, though, the GTP will be hydrolyzed, and we get deactivation of our G alpha. Now, one thing you may have noticed is that to get from our receptor alone to our ternary complex, our receptor's undergone quite some really big changes. We can see that we're now binding a peptide, but beyond that, we've got this kink here that allows our G proteins to bind. Now, each class of receptor does this differently, but they all use a series of conserved motifs through them. And these motifs have one job. They send a signal from the top of our receptor where the ligand is that it's ready to bind a G protein. And in doing so, it induces a series of conformational changes. Now, each class has a different series of these motifs. So I'm going to take you through class A and class B1 motifs just quickly. So here we've got a class A GPCR. It's adenosine 2A receptor. And I've highlighted TM6 for you down here because this undergoes our biggest conformation. In different colors, I've highlighted our conserved motifs for class A. So in orange, we have our CWXP motif, and this sits just below the orthosteric binding pocket for class A GPCRs. Below this, we have our PIF motif. Below that, we have the MPXXY, and down the bottom, we have the DRY motif. Now, these bottom two motifs are particularly interesting because these not only hold our receptor together in its inactive state, so keep it nice and tight together, but they also form interactions with our G proteins when they come along to bind. So now we're going to have a look at a transition between our inactive to active state and see how our motifs transform to allow our G proteins to bind. So we can see that as this transition begins, our TM6 starts swinging outwards, and our motifs rearrange to be able to support this new conformation and the binding of a G protein. And just to contextualize things, I'm showing the G proteins here. So in this sandy color, we have our G alpha S. In green, we have G beta. Our purpley thing is G gamma. And in black, we have nanobody 35. So we can see that this series of conformational changes that goes down our receptor is actually really important to allow that change to happen. When it comes to class B1 GPCRs, the motifs themselves are different, but the overall function is the same. So just below the binding pocket for the peptide, we have the central polar network, and this undergoes some changes when a peptide comes and binds it. Below that, we have the PXXG motif, and this motif is kind of interesting because it is coiled in one conformation in the inactive state, and then as it transitions to the active state, it unwinds and is held in a different position by a different set of hydrophobic residues. And down the bottom, we have the motif that's most important for my talk today. So that's the HETY motif, and this is located at the bottom of the receptor. It not only helps to hold our receptor in its inactive state, but it also helps to stabilize the active conformation and interaction with G proteins. And so we can see here, once again, we have a pathway that goes from the top where our peptide binds all the way down to the bottom where our G protein binds. And as we start to move to the active form, you're going to see one of the main differences here. And that's that our PXXG motif unwinds to form a much more dramatic kink than we were seeing in the class A GPCRs. This is something that's actually quite energetically expensive for our receptors to do. And again, we've got our G proteins down the bottom here. So these are Cryo-EM structures, and we've actually developed a bit of a sweet spot for Cryo-EM. So that's this ternary complex here, where we have our receptor, our G proteins, and our peptide all bound at the same time. And you'll notice maybe that I've showed our G proteins here without GDP or GTP bound. And this is actually really important because in Cryo-EM, we trap a really brief but high affinity state where the G proteins are really happy to be at our receptor. And you'll remember I said the role of the G proteins is to turn over GDP for GTP. So one key thing we can do is lower the likelihood that it's able to do that. And we can do this through mutations. So we have a bunch of different tools that allow stabilizations of our structures. So the first of these does just that. So our dominant negative mutations and our minimal G proteins both contain mutations and changes that disfavor the binding of nucleotides. So if our receptor can't bind nucleotides, it has no reason for the G proteins to leave. So the G proteins will kind of just happily stay there for a bit longer. And this allows us to isolate them for structure. We then also have stabilizing antibodies. Most popular is nanobody 35 and scFv16. And these stabilize the interface between our G alpha and our G beta gamma subunits. A final technique that's quite popular is tethering. I've shown here nanobit, but there are a range of different ways we can do tethering. And the main idea here is to have one of our subunits close to the bottom of the receptor and hold it in an artificially high local concentration to shift the equilibrium towards the complex. So today we're just going to talk about nanobody 35 and dominant negative GS. So if this is our Cryo-EM sweet spot and it's easiest for us to get structures here, how can we get to the structure if we don't have a ligand? And this is the question that underpins my whole thesis. And the answer I'm hoping we can use is called constitutive activity. But what is constitutive activity? Well, you remember our receptors exist in an equilibrium where we have our receptors and our G proteins and then complex on the other side. Constitutively active receptors have a naturally biased equilibrium towards this complex side. And this can be for a variety of reasons. It could be a disease-caused mutation, or it can be a natural facet of this receptor function. Now, these disease-causing mutations are interesting because they often exist within the motifs I mentioned earlier. And you can imagine this is because those motifs not only support activation, but they hold our receptor nice and tight in the inactive state. So if we weaken those interactions that keep everything together in the inactive state, we might make it easier for our G proteins to come along and bind and induce those conformational changes. So today we're going to be exploring constitutive activity in one receptor as my first aim. So the aims of my PhD were, on the face of it, pretty simple. So the first was to take the parathyroid hormone receptor and try to understand the structural impacts of clinically relevant mutations. And then the second aim was to apply what we've learned about constitutive activity and whether it can give us access to that Cryo-EM sweet spot by applying it to an orphan GPCR. So that's a GPCR where we don't know what the ligand is. So I'm going to begin by just talking about PTH1R. So the parathyroid hormone receptor, or PTH1R, is a class B GPCR. It's expressed all over our bodies. So it's in our bones where it's responsible for bone resorption. And this is one of the reasons there's a prime osteoporosis target, and there's currently 2 peptides which target it. However, it's also in both our kidneys and intestines, where it's responsible for calcium and phosphate homeostasis. It's activated by 2 endogenous peptides, the parathyroid hormone and the parathyroid hormone-related peptide. And today we're just going to be talking about parathyroid hormone and a truncated version of it. So most interestingly for us today is that we have a series of clinically relevant disease-causing mutations to PTH1R, which cause constitutive activity. Now, these mutations were first explored in the 90s, and it was noticed that they cause a rare form of short-limb dysplasia called Jansen's metaphyseal chondrodysplasia. There's about 30 cases diagnosed worldwide. And in the late 90s, Shapani did some studies investigating the pharmacological impact of these mutations. So these 2 mutations are histidine 223 to arginine and threonine 410 to proline. And so what she did is she took these mutations, these receptors expressing mutations, and did some cyclic EMP assays. So we can see here, this is the histidine residue. And on our x-axis, she's mutated this residue to every other amino acid. And on the y-axis, we're looking at the cyclic EMP response. So we're measuring the ability of our receptor to activate in the absence of any ligand. Now we can see that when we mutate to most amino acids, we don't see any changes. However, if we mutate to an arginine, which is our disease-relevant mutant, or a lysine, which is a very similar positively charged residue, we see an increase in constitutive activity. If we look at the second residue, we'll see something interesting. So this threonine 410, which if we're looking at the same sort of experiment, we can see that mutation to most of these residues will induce constitutive activity. However, today we're just going to be talking about the proline mutant, not just because it's disease relevant, but also because proline has some really interesting helix disrupting properties. And what's interesting to us today is the fact that these mutations actually lie in one of those key motifs of class B GPCRs that I mentioned earlier. So that is the HETY motif. And you'll remember I said it was down the bottom of the receptor and really important for activation. And that's because it not only holds our receptor together in its quiescent state when it's inactive, but also helps interact with our G proteins through water networks. And so we can see here, as we go from an inactive to active form, one of the residues, oopsie, oh no, one of the residues actually moves out with it. Sorry, we're gonna have to watch this bit again. One of the residues actually, so the threonine, if we have a quick look at him, we can see that the threonine in this motif swings all the way out as part of the unwinding of a class B motif. So if we look at the HETY networks itself and have a quick look at what the interactions are formed in both the inactive and active state of a class B GPCR. So it's called the HETY motif because it's a histidine, a glutamic acid, a threonine, and a tyrosine. And in the inactive state, we have two interactions. We have the histidine and glutamic acid, and the tyrosine and the threonine interacting. As we move to the active conformation, we can see we lose that interaction with the threonine, which has swung all the way out. However, we gain an interaction between this histidine and this tyrosine through pi stacking, and there's an interaction between glutamic acid and histidine here. So you can imagine we can disrupt this in two ways. We could stabilize this active conformation through mutations, or we could potentially try destabilizing what's holding everything together in its inactive conformation. So with that in mind, we have a few things we're looking for here in this section of my aims. We're first looking at whether we can find a Cryo-EM, we can access this Cryo-EM sweet spot with constitutive activity through these mutants. And then we're going to see if we can look at how these mutants are constitutively active. And we're going to do this through the structural models we can build from our maps. And this should give us a better understanding of how these mutations are causing organisms. So first of all, I'm going to show you some pharmacology. I did do this in my first year, but Alita recently confirmed it and did some wonderful work on it. So I'm going to show you Alita's pharmacology. So before we can go and, I guess, start just making things in insect cells and getting structures, it's important that we know what they're doing. So to do this, we've looked at these same constructs expressed in HEC293A cells. So to start off, let's look at the expression. And you'll notice that compared to our wild type, all three of our mutants have about a fifth of the expression. So there's a significant decrease in the expression of the mutants. And this is not uncommon for receptors when you mutate their key motifs. However, if we then look at the concentration response curve, we can see a few interesting things. Now on the x-axis, we have a concentration of PTH. And on the y-axis, we're showing the extent of the response as a relative percentage of the maximum response of the wild-type receptor. And so we can see, if we go to the maximum response, we can see that all three of our mutants have a decreased response compared to the wild-type. This perhaps isn't surprising because they have a fifth of the expression. However, the proline mutant, despite having a fifth of the expression, is actually fairly close to the wild-type. Whereas the two histidine mutants, which are also a fifth of the expression, are much decreased compared to the wild type. So I'm now going to show you the basal expression, which is actually already on this graph, but I'm just going to represent it a different way. So we can see here the response in basal cyclic AMP, so when we've got no ligand there. We can see we've got a significant increase in basal activity for the arginine mutant and the proline mutant, but not much happening for the lysine mutant. And I think it is possibly important to say here that it does look decreased. However, if we expression corrected, we may see some activity there for the lysine because it is a fifth of the expression. So now that we know that our receptors are doing something interesting, we know they have basal activity, and we know that when they're being stimulated by peptide, they can turn over more G proteins. We're ready to look at structure. However, I just wanted to briefly tell you how one gets a structure. So we start by expressing our constructs in insect cells. So rather than using mammalian cells, we've used insect cells, and we use baculovirus to get them. We'll express the receptor and the G proteins all together in one lump. And then because we've tagged our receptor with a flag tag, we can use affinity chromatography to drag out our receptor from its pellet. Then we can use size exclusion chromatography, so this is an SEC trace I'm showing here, to separate everything by size and get a nice pure sample. Then we'll often do some initial screening using negative stain. And this is really useful because you only need a low concentration of protein. And you can actually assess quite a few things. You can tell if your protein is dissociating on freeze-thaw. You can tell if it's homogeneous. So it can be quite a useful tool. And then once we're certain we've got something we like, we'll often move on to cryo. And typically this is for us down the road on the glacier. So that's a 200 kilovolt machine. You need usually at least 3,000 micrographs and over about 3 mg of protein, but it does depend. And once we've done a lot of different data processing, we get our Cryo-EM map out, and we're able to model into that coordinates which represent our protein and give us an idea of what's happening with our structure on a molecular level. So with that in mind, let's have a look at what we can do based on basal activity. So I'm bringing you back to this graph of Alitas. So in blue, we've got our wild type with no constitutive activity, our lysine, which potentially has a bit, our arginine with a lot, and our proline with a little bit less. And we're going to go through and see whether with the ligand and without a ligand, we can get a structure for each of them. So our wild type receptor here, it's not really surprising that when we have a ligand present, we are able to get a structure. And we can see that already from my SEC trace. So our first peak here is our aggregate peak, and this is followed by our complex peak, which is what we want. Our final peak is our receptor alone peak, and this is something we've seen fairly often with PTH1R. When I take away my ligand, I still see my aggregation peak, I still see my receptor alone peak, but I no longer see a ligand-free peak, a complex peak. And so this indicates to us that in the absence of ligand, there's not enough stability for us to get a complex. So no constitutive activity, no structure. And there are already structures available of PTH1R in the literature. This one was solved by Brian. It's 8FLQ, and it's the one we'll be talking about most today. So let's just step through each of our mutants now and see what we can get. So starting with the least constitutively active, the lysine, we can see that with the ligand present, we're able to get the aggregate peak, yay, and the complex peak. And then finally, what's interesting here is we don't get any receptor alone peak. And so this is a sort of hint to us straight away that maybe something different is happening here. So potentially, and it's hard to know without doing further work, but our receptor is less happy in its G protein-free form because of these mutations. Now, when I take away the ligand and do the same preparation, we'll notice something in our SEC trace. We still have our aggregation peak, and we don't have a receptor alone peak. And now we don't even have a complex peak. I guess if you're feeling generous, you could say maybe there's a shoulder there, and it is something I tried to optimize, but it wasn't successful. However, I was able to get a structure with a ligand. So if we move on to the next mutant, which has got slightly more constitutive activity, once again, we can see we've got our aggregation peak, our complex peak, but no receptor alone peak, just like the lysine mutant. If we overlay now our SEC trace of the ligand-free structure, complex, giving that away, we can see that we get a decreased yield, but we're still able to get some complex peak though. And correspondingly, I was able to solve a structure for both our ligand-free and our ligand-bound T410P mutant. Finally, let's look at our most constitutively active. So here we've got our ligand, our SEC trace with the ligand. We can see again, we've got an aggregate peak, we've got a complex peak, no receptor alone peak. And when we take away a ligand, interestingly, we get pretty much the same trace. And accordingly, I was able to get a structure of both. So this is already something fairly promising because it seems that with constitutive activities, we are able to get structures. So the next question then is, what are the impact of these mutants on the structures? So to do that, the first thing we're going to do is we're going to look at our peptide-bound mutant because we know we can still get a fairly good rate of G protein turnover with that. And we're going to compare each of them to the wild-type PTH1R that Brian solved earlier. So here in blue, we see our wild-type PTH1R bound to PTH1 to 34. And if I overlay our first mutant, the lysine, we can see that it's actually a very similar structure at a backbone level. We've got some differences down here in TM6, but the kink is overall in the same place. So what's happening at our location of the mutation? So in blue, we can see our wild type, and in our wild type, we get a bond between our glutamic acid and our histidine. However, the lysine, which I thought would be able to maintain that bond, is actually binding to the backbone carbonyl of a tyrosine. So although it has generally the same architecture on the outside and at a backbone level, we're actually seeing some differences in how or interaction at the mutation site. And so this was my least constitutively active mutant, and it also didn't have a very high Emax compared to the wild type. So next, let us look at the other mutation at that locations. This is the arginine, and we can see once again we have a very similar backbone structure. Everything overlays quite nicely. There's not a lot to write home about. However, if we look at the mutation site, we see some interesting things. This time, the mutation was actually able to compensate and forms a fairly similar interaction with the glutamic acid. It's not the same interaction, but it's compensating using a similar location. However, we have lost the pi stacking between our histidine and our tyrosine in the active state. So overall, we're getting a different compensation to what we saw with the lysine, despite them actually being fairly similar amino acids. So our final mutant is the T410P. And this was interesting to us for many reasons, but also because proline usually has helix disrupting qualities. And we can see here we've modeled the proline, and although I've modeled everything beyond there, it was actually quite hard and the density was really difficult to fit amino acids into. Now, if we look at the HETY motif, we can see straight away something that's pretty interesting. That's that this glutamic acid is completely flipped around. I did look for something else that it could be interacting with, but I couldn't find anything. Our histidine has also changed rotameric position. However, the proline, our actual mutant, overlaps quite well with the threonine. So overall, this was a fairly interesting structure to look at because we did see the weakness in the density beyond the proline mutation. However, at a backbone level, everything seemed to add up. So finally, if we overlay these structures, we see, again, with the peptide bound, they're quite similar to the wild type. So just to, I guess, summarize this first point, when they're bound to PTH1 to 34, our mutants are looking on a backbone level very similar to the wild type. However, when we get to the actual site of mutation, we do see some differences. So our lysine interacts with this backbone of the tyrosine, whereas the arginine, which is a mutation at the same location, is still able to form an interaction with the glutamic acid. Meanwhile, our proline mutant is forming no interactions at all. So overall, they're fairly similar. Now we're going to go through our mutants, both with and without a ligand bound. So in green here, I'm showing T410P with PTH1 to 34 bound. And in a minute, we are going to overlay the same mutant without a peptide. And we can see some fairly big differences straight away. So first of all, this kink that's really prominent in TM6 has shifted downwards. Interestingly, it's shifted downwards towards where our mutant is. Probably as a result of this, we see some changes propagating upwards. We see that our TM6, which is the one where the mutant is, TM6, 7, and 1 have all kind of shifted upright in relation to where they are normally. So now if we have a quick look at the level of motifs and look at the individual residues, we're just going to have a quick look at a morph between our ligand-free T410P and our ligand-bound T410P. And in this state, I want you to keep a close eye on the PXXG. So when our ligand-free, PXXG motif, which is usually where our kink happens, is all the way up here. However, as we move to our ligand-bound structure, it's going to shift downwards into its traditional location. To accommodate for this, our proline has to come back down to where we'd usually expect it to be if it wasn't mutated. So after we return to our ligand-free form, we're going to take a look under the hood and see what's happening with the motif itself. You remember that when ligand-bound, it didn't have any, we were seeing some interesting changes. So what's happening ligand-free? So again, we're looking at the HETY motif, and we can see in its ligand-bound form, we're getting this histidine here, which could maybe form a pi interaction with this tyrosine. However, this proline is in an entirely different location to what we'd expect from the wild type. The wild type or the ligand-bound mutant. So we can see as we transition into our peptide-bound T410P, we're going to see this proline swing all the way back out to where it was before. And then PXXG proline swings in to form that kink. So to sum up what we're seeing with our T410P mutant, we can see that when the peptide is bound, we get a very normal kink for a class B receptor. However, when we have a ligand-free structure, we're seeing something slightly different. It's less of a straight line than that indicates, but you can see the kinks in a different place. And this could potentially be reflected of the fact that when we look at our concentration response curve, we were able to get basal activity. However, it was nowhere near as much G protein being turned over as we saw when it was peptide bound. So perhaps it's assuming a different conformation that's able to turn over G proteins, just not as well as when we have the peptide there. Then if we look at actually just the mutation site quickly compared to the ligand-free and ligand-bound, we can see we have a few differences, the main one being the movement of this proline all the way out. So let's do the same thing with our arginine mutant. So we're just going to start with our arginine mutant as it is with the peptide bound, and we'll remember it's very similar to our wild type. In a minute, we're going to overlay it with our ligand-free mutant and see what changes there are. And we can see sort of straight away that a similar thing is happening. We see a movement of that kink and a change in where our TMs are headed. So if we then go and look at a similar thing, so we're going to look at a morph and how we change between our ligand-free and our ligand-bound form. We can see again our PXXG motif is up here in our ligand-free form, and the threonine is also turned inwards like we saw with the proline mutant. And as we transition to our ligand-bound form, we can see once again that this PXXG motif unwinds, and as it unwinds, all our TMs tilt over like we expect them to. And the threonine of the HETY motif has to come back out to assume that conformation we saw that's similar to the wild type. So there is quite a dramatic difference in that kink between our ligand-bound and our ligand-free mutant, which is something interesting already. Then if we have a look at our HETY motif, we're going to see some really interesting interactions that are happening with our arginine. So remember that our ligand-bound arginine was able to compensate for that interaction. However, our ligand-free one can do even more. This arginine, not histidine, is binding to the backbone tyrosine, this glutamic acid, potentially pi stacking with that tyrosine, and the glutamic acid is also forming an interaction with that tyrosine. So we've essentially got a lot of different interactions stabilized here. So our ligand-free form is actually fairly happy at that kink, I would say. And then we can see to move outwards into our active ligand-bound form. Ooh, hello. We can see that it did a lot of things. So again, if we overlay and just have a look at a static image, we can see that we've moved from a very stereotypical kink to something that's still a kink, still dramatic, but in a different location. And again, this might be a reason why we're seeing less efficient G protein turnover when we don't have a peptide bound. If we look at the actual site of mutation there, we can see what I was talking about before with our arginine in the ligand-free form forming more interactions, and so is that glutamic acid. Interestingly, though, is that this threonine is in a similar mutation as the proline the proline was ligand-free. And now you might be thinking that these two ligand-free mutants look fairly similar. However, if we have a look through the window here, we can see that our proline and glycine of the PXXG motif are actually in slightly different locations and they don't align perfectly. And this has potentially propagated up and resulted in the change we see over here in the black box, where we can see our two T-hems up here don't align very well. And then again, if we look at our actual motif itself, we can see that the arginine mutant was able to form some extra interactions where the proline mutant wasn't. And potentially we can think about this in terms of the fact that the arginine mutant was more constitutively active than the proline mutant. So perhaps this is one of the reasons why. So that's the end of the PTH1R story. So just to go back to this little chart, I do think the structures have told us how this mutation is causing constitutive activity. So if we go back to Alita's beautiful concentration response curve, we can see that we get this basal activity here, and we get this different TM6 kink. So it's capable of turning over G proteins, just perhaps a little less efficiently. And then if we fully activate our receptor with a peptide, we're able to get a similar conformation to our wild type, although there's some slight differences here. A further interesting thing we see is in our SEC traces back at the beginning. So you remember I said that we usually see this receptor alone peak when we do a PTH1R prep. We're not seeing that for any of our mutants. So this could potentially be something interesting to look at in an inactive state structure, which I will not be doing. And then this leads us to our second question. And that's whether constitutive activity can let us access the Cryo-EM sweet spot ligand-free. And it seems we are able to, based on the fact that we were able to get two ligand-free structures of PTH1R. So what happens now? What happens now if we look at an orphan receptor? And that leads me to my second aim. So orphan GPCRs are the 100 or so remaining GPCRs where we don't know their endogenous ligand. Just because we don't know what's activating them doesn't mean they're not interesting. And we actually have several drugs in clinical trials for orphan receptors that we don't know the endogenous ligand for. One of the reasons we may be having more difficulty adopting them, finding their ligand, is because they might be constitutively active. So they might be doing just what those PTH1R mutants are doing. They're able to form a complex without needing a ligand to bind. And they could be doing this in many ways. One way we've recently seen a lot of in the literature is a cryptic ligand. Another is part of the receptor acting as the ligand itself. So maybe ECL2 or the N-terminus dipping inside and acting like a ligand. So we have chosen GPR3. And so this is a really interesting receptor. I'm quite fond of it. And it's a class A orphan. It's GS coupled, and we believed it to be constitutively active. This is my pharmacology this time. And we can see that at an increasing DNA concentration, our cyclic AMP accumulation on the y-axis is increased relative to the control. However, it does maintain all those class A motifs I took you through earlier, except for the PIF. Physiologically, it's a very interesting receptor. So it's been implicated in Alzheimer's, in amyloid beta plaque deposition, but it's also been implicated in holding our oocytes in meiotic arrest. So it's got a role in premature ovarian failure, potentially. It's also involved in pain management and cocaine addiction, and has recently been investigated for a role in thermogenic lipolysis of brown adipose tissue. So it's a potentially very interesting receptor that we don't actually know a lot about. So we decided to try and pursue a structure for this, given that we knew it had constitutive activity and we thought this gave us access to structures. And I did this in a very similar way to how I did my PTH1R structures. So we started with co-expression in baculovirus and insect cells. We expressed the receptor and the G proteins at the same time. We then used affinity chromatography. And one thing you're going to notice straight away that puzzled me was that we see our aggregate peak. We see the second peak, which I thought was the complex peak. And then we see this third peak, which having done PTH1R, I thought was the receptor alone. However, I decided to collect both because better safe than sorry. We then took it to a microscope and looked at both of them. And we're just going to show our green peak and our yellow peak again. And we can see our yellow peak actually had a dimer. However, it wasn't a very interesting dimer. It had one G protein up this way, one G protein down this way. So it's not actually that interesting to us. However, for that reason, I'm now going to be talking about the second peak, which contained our structure. So we were able to get quite a nice structure, I thought, of GPR3 from our initial preparation. And I'm just going to take you through a couple of the things we saw. So straight away, we noticed that ECL2, which is sometimes what caused constitutive activity, wasn't really acting like a ligand. Instead, we noticed a series of a kind of weird sausage-like density that snaked from the top of our receptor all the way through to the transmembrane domain. And when we looked at the residues around this, we noticed they were really greasy. This was interesting because GPR3 had previously been proposed as an S1P, a lipid-activated receptor. And we found that we could actually quite nicely model a lipid in that top binding pocket. However, you'll notice there's some density left over. Now, in there, you can actually model a second lipid, or you could maybe be modeling one really long lipid. And so this was a really interesting question to us. Is our Cryo-EM showing us two lipids, and it's kind of just been averaged out through our processing, or is it one really long lipid? And to solve this, we talked to Tracy, who did some mass spec for us. And I'll be honest, I don't fully understand all the wonderful work she did. However, what she noticed is an enrichment of fatty acids between 14 and 22 carbon chains in length, and in particular, these 18 carbon chain fatty acids. So that includes stuff like oleic acid. And so, oh, hello. So if we quickly have a quick look, we can see that, is he going to work? Okay, never mind. We can see that all those fatty acids actually fit quite nicely into our density. And this led us to believe that GPR3 was not in fact constitutively active at all. And we published this paper, which described one of the ways that GPR3 is potentially lipid activated. So before we published, while we were in review, another two papers came out within a day of each other. So the first, oh my God. So the first one by Xiong and his colleagues identified oleic acid as the endogenous agonist of GPR3. Interestingly, this is also 18 carbon chains, just like we saw enriched in our mass spec. And then the next day, Chen, in the same paper, produced another structure that identified oleol ethanolamide as the endogenous agonist. This also has 18 carbon chains and one double bond. So they're very similar fatty acids. However, they have different head groups. So this is our oleic acid head group. And over here, we can see we have a different head group for, I'm going to call it OEA, because I'm not saying that name again. And so what we decided to do is go and get a structure to see if we could see any changes if we added in extra oleic acid, a potential ligand, into our preparation. So I did the exact same thing. I expressed everything in my insect cells, and then I got my SEC trace. And you can see if we overlay our previous SEC trace, we've seen a reduction of this void, so a reduction in aggregation, a decrease in the dimer, kind of, and then an increase in our monomer state. So this was a pretty promising SEC trace. And we got about 7 mg per mil, which is an all-time high for me. And we took it to the Krios, and we managed to get a really nice structure out of this. And even from my local resolution map, you can start to see the lipid point poking through there. And you can see actually the second lipid down the bottom in 2 potential conformations. And so if we overlay, so here we're looking at my old structure, the one that we published in biochemistry, and I've got that in cyan. And then I'm going to overlay our new structure, which is the one we published in the presence of excess oleic acid. And we can see that I've modeled that extra, those 2 extra oleic acids, and it's up. We found density for the fatty acid a bit further up this time. One of the more interesting things we found was that with our better resolution this time, we could model ECL2 a bit better. ECl2 is really dynamic. We can tell from our variability analysis that it's moving around a lot. And something that's kind of interesting is none of the 4 structures actually agree on how to place a model ECl2. So it's probably quite dynamic. Another interesting thing is this histidine and this tyrosine. Now, both Chen and Song did some mutagenesis studies, and they found that if you mutate one of these to an alanine, either one, you get a reduction in signaling. And if you mutate both to an alanine, you get no signaling. And so they believe that these 2 residues are quite crucial in the interactions, and they've actually modeled a water network connecting them. I couldn't see any water. However, it is interesting to note whether I added extra oleic acid or relied on the oleic acid in cells, those residues do seem to be in a similar rotameric position. And so this actually wraps up most of my stuff today. So just to conclude this final bit, we were actually able to show that if we have high basal activity, we are able to get a structure quite easily. And then by adding extra oleic acid in, we were able to show that GPR3, whether or not it endogenously is activated by oleic acid, it at least can bind it and we can get a structure with it. I'm briefly going to go through some future directions. So I'll take you all the way back to PTH1R and this basal activity scale. And you remember I said lysine was the least constitutively active and we weren't able to get a structure. However, I only used 2 of the very many stabilization techniques I mentioned. And you'll notice that even with 2 of them, we still get a small peak that could potentially be optimized. So what happens if we're a bit more aggressive with our stabilization, if we use something like nanobit tethering or tethering RGS? Could we potentially get a ligand-free structure? So this could be important for our scaffold in that whether you have a little bit of constitutive activity or a lot of constitutive activity, you could potentially still get a structure. You just might need to give it more support to get there. There's also something interesting to be looked into with our peaks when we do and don't have those mutation presence. And this could be linking back to some inactive states. And perhaps our mutations are actually destabilizing some inactive interactions, the HETY motif. And this would be very interesting to get an inactive state structure of. Relate our scaffold in a receptor where we know a fair bit to our orphans where we don't know everything. So I've represented here an orphan receptor. And you'll remember I said we have these equivalent motifs in class A GPCRs. So what happens if we mutate some of those and we can potentially get constitutive activity? So perhaps we could look at doing something similar to what we did with PTH1R. We can get an orphan receptor with no constitutive activity, try engineering some in, and see if we can get a structure that way. And this could be a potential strategy for getting orphan GPCR structures. Finally, we had our paper, we published that, but there's also this paper with oleic acid, and we've shown that you can get a structure with extra oleic acid in. However, there was the paper published a day later about OEA. So can GPR3 bind both of these? If it can, are there different interactions at the head group? Why can GPR3 bind 2 very similar molecules? I think there's some really interesting questions there to be talked about the promiscuity of GPR3 and the activation mechanism. So that's it for me today. There's a lot of people I'd like to thank because obviously you don't do a PhD by yourself and you don't get that many opportunity to thank the people in your life, actually, I've realized. So everyone gets a very sappy thank you from me today. So first of all, to Patrick, my primary supervisor. Patrick has been a constant source of support for me throughout these 4 years. In my first year, he very kindly sat me down and explained how G proteins actually work because I was very much not understanding everything that was happening, and I'll never forget that kindness. Denise has also just been a very lovely, kind, constant support throughout this whole process. It's been a very long 4 years, and starting in COVID is no one's best idea. Cindy, who is hopefully online because she is actually currently pregnant. I'm extremely, extremely lucky to have Cindy as a supervisor. She has put up with so much from me. If you don't know, I frequently confuse micromolar and millimolar, and I've given Cindy several heart attacks, and she has been very patient with me throughout the years. I came here not really knowing how to pipette anything. I went through a phase where I contaminated my cells just non-stop, and Cindy has just been a lovely support through all of that. She taught me how to do preps, taught me how to pipette. I'll be forever grateful. And if she's anything as good at being a mother as she was a supervisor, she's going to be pretty good. My final thank you on the supervisor side goes to Matt, who he's taught me not to break a microscope, and he's taught me how to do, or at least understand all the math that's going on, even if I could never reproduce it myself. And just been an amazing support, especially in the past writing phase, which is so much fun. I'd also like to thank my industry partner, AstraZeneca. I had a wonderful time on placement, and I really valued the time I spent there. Well, sorry, I should say, this is Cindy, my supervisor, and this is Elaine, her first primary student. Cameron and I were her first students, but Elaine's her first student as a primary. We're all very proud of Cindy and everything she's accomplished. These are my two supervisors from AstraZeneca, Gavin and Fiona. They're both amazing people and wonderful scientists, and I really enjoyed my time there. I'm also grateful to my industry partner for our monthly meetings at ungodly hours. I'd like to thank my panel members, Nat, Alyssa, and Celine. They were very, very kind people who had some excellent pointers along the way. In particular, Nat has been an amazing mentor. Jackie and Tracy, you have had the unenviable task of wrangling academics, and you do it very well. And I wouldn't have got to England if it wasn't for Tracy, so thank you very much. The entire group, you're wonderful to do science with. In particular, Sam taught me how to not infect my cells, which is a really useful life skill. And Sam, Oliver, and Elliot, who's moved to Denmark? Moved overseas. We're all very useful in that first year. So yes, thank you everyone in the metabolic receptor biology group. I would like to formally apologize to anyone who had to sit near me in a desk. I'm really loud. And thank you to the Ramachodiot Centre and the Ian Holmes Centre and Massive for all this support. This is my wonderful family. We all look really similar. It's my mum's side, that's mum there. I've been incredibly lucky to have their support from Sydney. And finally, my wonderful friends. If you don't know, I live with this amazing little girl, Ivy. She is just wonderful. Nasuma, Montana. And I've made a lot of wonderful friends while I've been here. It's been a really fun 4 years. So thank you.
There is a little reminder that you actually spent time here.
Oh, thank you. Awesome. Will I get a thesis too at the end? Or?
It's not finished yet either. Nearly done.
Sure.
All right. So thank you. Excellent seminar. Good coverage of an exciting project. Open up the questions. I don't know if anybody's monitoring those online. But...
Oh.
Any questions?
No one has questions.
Yeah, we can. Let's go.
Well done, so thanks.
Thanks.
Congratulations. I had a question about your PTH mutant structures.
Yeah.
Obviously that DM6 is in quite a different conformation.
Yeah.
Does that alter the G protein interface in any way?
Oh my gosh, what a great question, Cameron. And yes, it does. So so we've got our wild type here, and we see, I'm not very good at describing G protein interactions, but I'll do my best. We see a general reduction in the amount of contacts. Also, interestingly, for the arginine mutant, we do see a van der Waals interaction with the alpha 5 helix of our GPCRsI. I don't know, of the G alpha subunit. So there is a difference in those interactions, and it definitely decreases. Decrease interactions.
Alpha.
Really, really amazing work. Congratulations. That asked my previous question. I'll change to another one.
Awesome.
It's going to be more pharmacology. So for your mutation,
yes.
When you saw AD, so is the T410, T41, T410P. That one has lower cell surface expression. It does not seem to be maintained very well.
Yeah.
Do you know whether the total protein production of that mutation is similar, or whether its proportion delivered to the cell membrane is reduced, and whether that mutation can signal from intracellular?
Okay, so PTH1R can signal intracellularly. We have not measured that specifically. We've just measured the cell surface expression and the total cyclic AMP production, but it is a very worthwhile question, especially given that we do know PTH can signal from endosomes, but I haven't measured it.
Snowfly.
Sure.
Great talk, by the way.
Oh, thanks. Congratulations on the fellowship. Yeah. Good job, everyone.
So obviously you have different protein interface, but then
Yes.
you also have reduced response to the ligand, right?
Mm.
And so you mentioned that it's maybe because you have different exchange for the GTP or GDP on the G protein. So the question is, right. The question is whether your G protein have different conformations in your ligand-free versus constitutive activity, and if not, whether you or somebody else have tested kinetics of G protein linkage or exchange.
So the G proteins themselves do align fairly well. We're obviously seeing these differences in interaction. So we see a decreased interaction across them. The lysine mutant, least constitutive activity, least interactions between the G protein and the receptor. Kinetics wise, I have not done those studies. I believe Elite has done some true path, but we've not got the end numbers up yet. So definitely more work to do there.
Great work. So now that we talked about the G protein interaction, what about the ligand? Ligand interactions.
Question, Fabian. The G protein, the ligand interactions do decrease. There are. So PTH1R on the wild type makes a lot of interactions. Generally, you do see these decrease. Proline mutant has the least amount of interactions. You do see a general decrease. So here I've got in bold are the interactions that are conserved across all of them. And then if it's italicized, it's an interaction between two or more. So decreased, yes, I haven't found any pattern, but I'm sure there is one. I'm sure I'll be told about it in my feedback if there is. Matt?
I don't know if I'm allowed to ask. Am I allowed to ask? Izzy, bloody amazing talk. Now, you and I have discussed this a little bit, but what do we think? Is it possible to deorphanize GPR3? Or do you think it's just a promiscuous fatty acid activation sort of process?
So we know that GPR3 is upregulated in cold temperatures in brown adipose tissue. We know there are ways we can increase GPR3 expression based on like environments like that. So I think potentially it's a very promiscuous receptor that's upregulated in certain scenarios. And it doesn't really matter what's there potentially, as long as it's roughly the right length.
So the follow-up question is, what should we, once we deorphanize it, what should we call it?
That's a great question. The Parkville receptor. I don't know. MIPS.
Russell receptor.
Sorry, I stole your job. You do your job. No, that's just my family.
Okay, Laura.
Fantastic talk, really enjoyed it. So my question is kind of on the future direction, the things that you kind of got a bit excited about. So which orphan receptor, apart from GPR, would you really like to try the linkage on?
There are a lot. There are like 100 orphans. There's plenty you could do. I also think it would be really cool if you could get we could try another orphan that did have constitutive activity. So maybe GPR21 or something. I think Dong-Ju's receptor actually would prove pretty cool to try mutating, because it's proton sensing as well, I believe. So that could be interesting to try mutating and see if we can get access to it a bit more easier. I think that's 151. But yeah.
Response to this question. And you think it can be a transporter for the folic acid?
Yeah, potentially. Yeah, so we do have some evidence from 3D-VA that it's actually sort of shuttles the lipid through the receptor through a sort of twisting motion of the TMs. So it could have a role like that. I think it could definitely partition lipids through the membrane, but I'm not too sure. It's a possibility.
Just follow up on this previous question.
Yep.
The structure you're using dominated.
Yes.
Yeah, and with institute activity or active researcher, have you thought about or whether it is possible to use wild-type genes? And just fulfill the pattern.
We can potentially. We can definitely get structures with wild type. It might be a bit more difficult just biochemically to make it stick on because it can still exchange those nucleotides, so it will be wanting to leave the receptor, but we could definitely try and we might see some interesting differences there.
Have other questions? Yes.
So back to the GPCRsI.
Yes.
So can you have protein inside organelles? And if so, why wouldn't the diamond be interesting? Because then would it potentially be happening intracellular?
I suppose it could potentially be happening intracellularly. That's a good point. I just hadn't considered it before. We didn't get very far with the structure because I did try processing it, but it was kind of difficult. But it wasn't as interesting to us as the monomer. But yeah, definitely if it was in like an endosome or something, it could still be useful.
Do we have any other questions? We've had lots. If not, I'd like to again thank Isabella for a really wonderful course. Nice one.